How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hello and welcome to DNA Today. I'm Kira Deneen. DNA Today informs you on what's happening in the genetic world. You can tune in every Monday at 11:30 a.m. to WHUS.org or 91.7 FM. You can listen on your own time over at DNApodcast.com. I have all the episodes on there. Today is a very special day. We are celebrating DNA Day, and so this is obviously a big deal, especially for the show called DNA Today. It's actually DNA Today. So what is DNA Day? Well, National DNA Day commemorates the completion of the Human Genome Project in April 2003 and the discovery of the double helix of DNA in 1953. So this year's DNA Day is now Monday, April 25th, 2016, and it began to be celebrated annually on April 25th after the 108th Congress passed concurrent resolutions designating it as DNA Day. So it's a national wouldn't say holiday, but it's a national recognized day. And the goal of National DNA Day is to offer students, teachers, and the public an opportunity to learn about and celebrate the latest advances in genomic research and explore how those advances may impact their lives. So to celebrate on the show today, I'm going to be talking about what DNA is, because people probably hear of DNA, but do you actually know the structure? Do you actually know what goes into DNA? Also talking about the history of humans discovering DNA and the new aspects of DNA, and we're constantly learning new information. This is a exploding field in science. Also going to be talking about the fun events you can take part in today and the careers that relate to DNA. So everything is DNA today on this episode. But before I get into all of this fun stuff, I want to ask you guys for a huge favor. The Podcast Award nominations are happening right now. So if you like this show, please go to podcastawards.com, scroll down and find the category Science and Medicine on the right side of the page. Fill out the show name as DNA Today, and the URL is dnapodcast.com. Scroll down, fill out your name, email, hit submit. That's all you have to do. Comments are welcome if you want to include any, but really looking for you guys to help me get nominated. Again, podcastawards.com, Science and Medicine category. The show is DNA Today. The URL is dnapodcast.com. All right, and now for this fun stuff. So starting out with what DNA is. Obviously, DNA is a very big deal. We hear about it all over the place. It's the blueprint of life. But what is the molecule DNA? Well, DNA stands for deoxyribonucleic acid, and each cell has different functions it needs to carry out in order for the body to be able to function properly. For instance, a cell in the liver will differ from a cell in the heart, and DNA is responsible for this difference, but DNA is the same in both those cells. It's the proteins expressed from that DNA that differ and make the cells different. So DNA is identical in all of your cells, assuming you're a healthy, normal human. There are so many different things that can go wrong to affect that, but most of us have the same DNA in all of our cells. You're probably familiar with DNA's shape. It's quite iconic as it looks like a twisted ladder, and this twisted ladder shape is referred to as the double helix. A ladder has two parts, the sides and the rungs. In DNA, the sides of the ladder are made up of sugar phosphates, while the ladder rungs are made up of 4 nitrogenous bases. So these 4 bases you've probably heard of, A, C, T, and G. So we have this ladder, we have the sides of the ladder, sugar phosphate, and the rungs of the ladder, the nitrogenous bases. 2 of these letters are per rung. A only pairs with T, and C only pairs with G. So A with T, C with G. A and G are purines, and T and C are pyrimidines. That talks about the structure of these nucleotides. A and G look a little bit more alike compared to T and C. So pyrimidine pairs with a pyrimidine. Purine pairs with a pyrimidine. So we have A pairing with T and G pairing with C. In order for these letters to stay connected, in order for the rungs of the ladders to be stable, they're bonded to each other. And the bond that holds these two letters together, which, again, we're talking about the rungs here, are called hydrogen bonds. And they're a little bit easier to break, which is important when we're talking about DNA replication, where that DNA helix gets unwound a little bit. A pyrimidine is a single-ringed base. So this is going back to talking about thymine and cytosine. That's what the T and C stands for. And those look similar because they only have one ring in their base. Whereas a purine, the adenine and guanines, the A and Gs, have a double ring base. So they look more similar than the T and the C, the thymine and the cytosine. So let's go with an analogy to help us understand what the nitrogenous bases do. So we have all these letters, right? And they're all pairing together, but how does the DNA end up having such a complex molecule if it's only using 4 different nitrogenous bases? We have 4 letters. And when they combine, they form words which then form sentences. These sentences make up the genetic code, and that's what we call genes. The number and order of the nitrogenous bases determine what proteins are made, and that's how we read it, and that's how the cell reads it. Just like the number and order of words determine different meanings, like cat is different from tac, C-A-T versus T-A-C. Even though they have the same letters, the order of the letters makes all the difference, like when we're reading. One strand of DNA contains so many different types of genes, and each cell in our body contains enormous amounts of DNA. In one cell, if you pulled out the DNA and stretched it, it would be 3 meters long. And just to put this into perspective, this is about as long as a car in every single one of your cells. So that just by itself is mind-blowing. So in order to fit all this DNA into one tiny, tiny little cell, chromosomes are used. This is when the DNA wraps around proteins, and these proteins are tightly packed until they form this molecule, this little complex called a chromosome. And with all these steps, chromosomes are very efficient in storing data. We have 46 chromosomes if we're a normal, healthy human, and these pair off into 23 pairs. You get 23 pairs from your mom, 23 from your dad. And that's a little bit of a look at DNA. We're talking about the double helix structure. We have the adenine, cytosine, thymine, and guanine making up this DNA language. And in order for these massive amounts of DNA to be stored, it's usually all kind of bundled up into what kind of looks like spaghetti. But when the cells actually replicate and it goes through cell division, that's when those chromosomes are composed. And the proteins in DNA are wrapping around each other in order to form these chromosomes. A little history of DNA. So you're probably familiar with Watson and Crick. You've heard their names. They discovered the structure of DNA. But DNA's history goes back further than just Watson and Crick. It goes back further than 1952, which is when Watson and Crick discovered DNA. All the way back, and this is an abridged version of the history of DNA. Obviously, I don't have enough time to go through the entire history, so I pulled out some interesting points and years that we made strides in discovering more and more about DNA. 1869, all the way back in 1869, DNA was first isolated by Frederick Miescher from pus in the bandages of wounded soldiers and fish sperm. So that was the first time when DNA was isolated, when we were like, this that we pulled out here is DNA. In the early 1900s, Thomas Hunt Morgan, another name you should be kind of familiar with, Thomas Hunt Morgan proposed that each chromosome, remember that's when the DNA is more condensed with those proteins, each chromosome contains a collection of these small units called genes. And this is a term he adopted from the Danish psychologist Willem Johnson, who had lectured in Columbia in 1909. And from that, he was able to, he took that term genes and kind of applied it to thoughts he already had. And he also was the one to think that with different genes that they have specific locations along specific chromosomes. And that's something that's very key today, especially with diagnosing patients with different diseases. 1938 was when we found evidence that DNA is a polymer consisting of many bases. Those are the A, T, C's, and G's that I talked about. 1944 is when Avery and his colleagues showed that DNA carries the genetic code. So it wasn't until 1944 that we understood that DNA was the thing that was the hereditary component of the cell. We thought maybe it might have been proteins before this, but Avery's work and followed up by Griffith's work in London later really established that it is the DNA that carries the genetic code and the inheritance patterns that we see from parent to offspring. 1948 was when Chargaff found a pattern in DNA's chemical letters. So before this, we knew the guanine, cytosine, adenine, thymine. But in 1948 is when Chargaff found this pattern of DNA saying that the amount of guanine equaled the amount of cytosine, and the same relationship for adenine and thymine. Now we know this is because those two pair together. So if there is 20% guanine in a certain gene, say, there's also going to be 20% cytosine because they're partners. And this is where things should be super familiar. In 1951, Rosalind Franklin created a high-quality X-ray diffraction photo to reveal more detail of repeating structure of DNA. And 2 years later, 1953, Watson and Crick discovered the structure of DNA, having that double helix, very iconic look, based on Rosalind Franklin's photograph, which her name was not included in the Nobel Prize that Watson, Crick, and another scientist, a colleague of Rosalind Franklin, Maurice Wilkins, they got the Nobel Prize for it. But Rosalind Franklin did not get credit for this. And I did a whole episode, actually episode 2, all the way back to episode 2, about Rosalind Franklin and how we can't forget Rosalind Franklin. So you can go back to DNApodcast.com to listen to that episode. Really interesting to kind of read about everything that she did versus the work that Watson and Crick did and how they just kind of expanded her work. Going along with our history of DNA, 1961, Jacques Monod and Franz Jacob showed that messenger RNA helps turn DNA into a protein. And this is when we started to establish the dogma of genes turning into proteins. So we have DNA going to mRNA, stands for messenger RNA, to proteins. So in order for proteins to be made from DNA, it needs this kind of middleman in the process, and that's messenger RNA. 1973, genetic engineering was born, and today, the last 3 years, this has really exploded with a technology called CRISPR, and that has a lot of high potential for medicine and treatment, and just editing genes, which is just really, really cool technology. It's a little bit hard to understand CRISPR, but it is definitely a really state-of-the-art technology that is currently being developed. 1977, Sanger presented a rapid technique to read DNA, to sequence the bases of the genetic code so that we could say, all right, we as humans can read what this DNA is saying. This technique is still used in labs. Sanger sequencing is what it's called, but it's slowly being replaced by next-generation sequencing techniques. There's a lot of different generations of next-generation sequencing, and that one is kind of taking over labs, but Sanger sequencing is still used today, and it was developed in 1977. 1983, Kary Mullis, very interesting guy. He invents the polymerase chain reaction to amplify DNA, which is now crucial for genetic testing. I have done PCR before. Pretty much any genetic student, undergrad, or even sometimes in high school has done PCR. Very common to do, and there's an interesting story behind Kary Mullis. Super interesting guy, and he was the one that invented this PCR, and he was actually driving on down the highway when he thought of this idea. He wasn't working in the lab, he wasn't, sitting there trying to think of something. It kind of just occurred to him. So that's kind of an interesting angle from this, because usually all these discoveries are made in the lab, but he's a very creative guy, and he was just driving down the highway, and it kind of occurred to him as the lines on the highway were morphing together. He kind of thought of PCR and how he could amplify a segment of DNA so it was easier to look at. Next year, 1984, the Human Genome Project was started, and DNA Day is celebrated because in April 2003, that's when the Human Genome Project ended, and it took 20 years, really long time to go through it, and it cost massive amounts of money, but definitely worth it. And when the draft was completed, again, 2003, Clinton said, "We are learning the language in which God created life. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal, and we're still doing so." So that is a rapid, quick round of DNA history, and it really is just the surface of how we've started in genetics and where we are today. And there's even more that has happened since the final draft of the Human Genome Project. And the timeline that I shared was published on The Telegraph. So if you're interested in reading much more about it, you can go to The Telegraph. So there are lots of different things that are going on today to celebrate DNA Day. I participated in one event this morning, which was the tweet chat, National DNA Day's hashtag DNA Day 16 Twitter chat, or tweet chat. Most people say tweet chat. It was moderated by the handle DNA Day, and they posed questions to programs to involve genetic and genomics research and education about topics like career paths, technology development, education. So I was able to participate in the first half. The other half is actually going on right now, so you can pop on to Twitter and just search hashtag DNA Day 16. And they asked participants to define gene at an 8th grade level, share a favorite gene. I shared the FOXP2 gene, which is really cool. It's critical for our high verbal skills and sets us apart from other primates. Other primates do not have this gene. So that's really interesting to see how we're different from other primates, and this is one of the genes that kind of differentiates us. There aren't actually that many, which is interesting. Another question they asked was what is the best model organism for an animal-plant study to simplify biological systems? And I chose mice because we share many genes with them, and they're a little bit easier to breed and give homes to than primates. Primates would be more advantageous to study because they're even closer to us genetically, but mice are actually pretty close to us and they breed a lot faster. If you missed the tweet chat, you're interested in reading transcripts, you can check out archives of previous tweet chats and other online discussions from DNA Day. I'll put the link in the show notes. They said they would provide they've provided old archives of episodes, or not episodes, that would be me, old tweet chats, and they will be putting today's tweet chat up very shortly. So it's definitely fun to read, and it's interesting to see who participated and kind of looking at their background, how they're involved with genetics. So that one already happened, but there's a couple that are upcoming. Every day last week and today, in celebration of DNA Day, a different expert has been answering questions on Reddit, Science Community Forum, which is /r/science. That's reddit.com/r/science. And the National Human Genome Research Institute, which is kind of leading a lot of these events happening, and the American Society of Human Genetics are joining forces to bring prominent geneticists from a variety of fields to Reddit, so you can ask them anything. This is a common thing done on Reddit where someone is answering questions and people just kind of ask them anything that they're thinking, anything that they're curious about. Today's guest on Ask Me Anything on Reddit is Dr. Francis S. Collins. He's the director of the National Institute of Health, and we'll be answering lots of questions today. And in that role, he oversees the work of largest supporter of biomedical research in the world, spanning from the spectrum from basic to clinical research. He's a physician geneticist noted for his landmark discoveries of disease genes and his leadership of the International Human Genome Project, again, something we're celebrating today on National DNA Day. He served as the director for the National Human Genome Research Institute at the NIH from 1993 to 2008, so he definitely has a lot of knowledge under his belt, and any kind of genetic question you have, I bet you he can answer. So go to reddit.com/r/science to hop on there and ask him anything. So something that's very cool to me, and I'm very excited to be tuning into, is a National DNA Day speaker series. And today's is a presentation on kind of pop culture and genetics genomics. The guest speaker is Eric P. Spana, and Dr. Spana's discussion topic is, get this, Harry Potter and the genetics of wizarding. I'm not sure exactly how that's all tying in. I've seen a couple articles and things that talk about if the wizarding world was studied, this fiction wizarding world in the Harry Potter series by J.K. Rowling, if it was studied, how would the inheritance patterns of being a witch or wizard work? I don't know if that's the topic, but I kind of have a guess on that. So really interesting. Definitely not a typical genetics webinar that you would usually attend. It's taking place from 3 to 4. I'm guessing that's Eastern. It didn't specify, but I'm guessing that's Eastern time, 3 to 4 p.m., and I will put the link in the show notes, dnapodcast.com. I'm not sure the exact link yet, but I will be putting that in the show notes. And Dr. Eric Spana is an award-winning instructor. He spent over 20 years utilizing Drosophila, fruit flies, as a model system for neurobiological cell biology and genetics. He's got lots of experience, and he claims that he's a fan of Harry Potter, Star Wars, and the Avengers. So he seems like a pretty cool guy. If you're interested in finding out what he's going to be talking about in terms of the genetics of wizarding, that starts at 3. I'll also be tweeting out the details too if you follow on Twitter at DNA Podcast. And that's about it for the events online, but there are local events. You can also find events in your local area using a map locator. So in CT, there is an event in Middletown called Strawberry DNA Extraction Workshop for Discovery After School program. So some of you listeners may be attending that. It's hosted by the Green Street Teaching and Learning Center of Wesleyan University. Unfortunately, this program is not open to the public, but other events are. The closest I could find to Storrs, Connecticut, where I am at UConn, is Brown University. Brown University is hosting DNA Day with SciTunes in Providence, Rhode Island, and it starts at 6 tonight, so you still have time to plan and go to that. I don't really know much information about it, but you can learn more by contacting the event coordinator at SciTunes. That's SCITOONS DNA Day at gmail.com. I'm sure they can answer all of your questions on there, and it seems like a pretty fun event. And there are so many events online that you can be participating in. Lots of fun stuff. Definitely recommend that Harry Potter one because who knows what he'll talk about. I'm sure it's going to be interesting. So what are some of the careers you can do with DNA? Obviously, I keep saying this is a vastly exploding field, but if you're interested in joining the field career-wise, how can you get involved? What are the opportunities out there? First and foremost, I have to plug genetic counselors. I aspire to be a genetic counselor, and genetic counselors are a healthcare professional with specialized training in medical genetics and counseling. They specialize in prenatal, pediatric, hereditary cancer, cardiac, as well as other fields. The cardiac field is a little bit more new compared to the other fields, and they basically just sit down with patients and go through their genetic history with them, their family history, and based on that, they can help guide them through genetic testing, explain more, point them to support groups and things of that nature. Cytogeneticists, this is what I am currently getting a degree in. Cytogeneticists study chromosomes, which are the structure of DNA. So chromosomes, that's what I talked about with the DNA wrapping around proteins to be really condensed. And when they're in this shape, we can actually identify which chromosomes are what, and we arrange them in a karyotype, that's what it's called, and we start out with the biggest chromosomes, moving down to the smallest. There's 23 pairs. The only thing that is the exception to this big to small rule is our sex chromosomes. Those go at the very end. The Y, which is found in males, is the smallest chromosome, and the X is similar to the size of like a chromosome 6. And this definitely takes a trained eye. It's very frustrating at first, but it becomes fun and kind of like a puzzle when you're going through it. And as cytogeneticists analyze these karyotypes, so they have a metaphase. If you remember back to 10th grade biology, you have the cell cycle, and part of that cell cycle, when it's dividing, is a metaphase. When it's metaphase is when we can actually see these chromosomes. And so we stop cells in metaphase, then we can take pictures of them on computers. And then on a computer, we can do this karyotype. And once the karyotype is done, arranging all the chromosomes and organizing them, we can analyze the chromosomes for abnormalities. We can see if there's an extra chromosome. That'd be a trisomy, such as trisomy 21 Down syndrome. If we are missing a chromosome, monosomy, if we have a deletion, if we have an insertion into a chromosome, if we have a translocation between chromosomes, there are lots and lots of different types of genetic abnormalities, and that's a whole episode on its own. I could go on and on about all the different types, but it's really, really cool field. The other type of job position that my program trains for is molecular diagnostic technologists. They analyze biological markers. So instead of looking at chromosomes, they're just looking at the DNA and breaking it up in certain ways in order to look at these biological markers in the genome and the proteome. And they use techniques such as next-generation sequencing. That's what I mentioned. That seems to be the technology that's being used today, and kind of taking over Sanger sequencing. And this sequencing is a way of sequencing our DNA and being able to read it. Also using polymerase chain reaction. Again, Kary Mullis, the guy driving on the highway, is the one that invented that. Gel electrophoresis, and many more different types of techniques. A couple more different careers that you can do with genetics is being a medical geneticist. You're actually a full-blown doctor, and their job is to explore human genes and discover new information about the function of each one, as well as the effect it has on the human body. So that one is a little bit more of a broad job in the sense of kind of what you get to do and who you are affecting indirectly and directly. Forensic scientists seem to be a field of interest, especially for kids watching CSI and shows like that. They are commonly known for investigating crime scenes, but their job also requires analyzing the DNA that they may collect and completing vast amounts of laboratory research. So that's a really interesting field, and a lot of kids, hear forensics and they get really excited. So that's definitely a really cool field to go into. And one that you may not think of is physical anthropologists. They work in environmental settings, searching for evidence of ancient existence and studying human evolution. So really looking at evolution and finding samples, ancient samples, and deciphering genetic codes and finding biological variations between humans and primates, both past and present. Those are just a couple of the different careers that you can do within genetics. There are so many more. I just mentioned like 6 here. There's so many more to look into, and it's even hard to find a list of all of them. So obviously the field of genetics and genomics is growing so fast. There's never a dull moment in the news as advancements are constantly being made, especially with personalized medicine, CRISPR-Cas9 technology, that's the gene editing technology I mentioned, cancer research, rare disease treatments, cures, so much happening. So if you have any questions about DNA Day and its events, you can direct them to dnaday@nih.gov. You can also learn more on their website, genome.gov, and they have links on there to get to the National DNA Day information. If you're a fan of the show, please help nominate me in the Podcast Awards. You can go to podcastawards.com, scroll down, find the category Science and Medicine, and then you can type in DNA Today as the show name, dnapodcast.com. Scroll down, put your name and email in, hit submit. Thank you so much for helping me get nominated. So happy DNA Day. Thanks for listening, and join me next week, again Monday, 11:30 a.m. to learn and discover new advances in the world of genetics.